Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the unit of life, and today in this video we will cover part 3. So people, those who haven't gone through the earlier parts, part 1 and part 2, so have a go through. Okay, now let's begin with the first question here. Now this question was asked in the year 2015. The function of gap junction. Okay, so what is the function of gap junction is the question. So what is gap junction? You have to know what is gap junction first, right? They are actually specialized intracellular connection between the multitude of an animal and it can be of different types. It depends upon the cell types. They directly connect the cytoplasm of two cells, which allows various molecules molecules, ions or electric impulses to directly pass through a regulated gate between the cells. So this gap junction will facilitate the communication between the adjoining cells by connecting the cytoplasm. Okay, so as I already said, a gap junction is a specialized intracellular space. Okay, it's an intracellular space which facilitates the communication of various molecules, ions, electrical impulses. The molecules can be different. It can be smaller, larger, transfer of ions, everything. So what does a gap junction do? It will facilitate the communication between adjoining cells by connecting the cytoplasm for rapid transfer of ions, small molecules and small large molecules. Okay, so therefore the correct answer to this question is option A. Now if you look here, I have given you a picture to your right side which shows this gap junction here. Okay, so it's between the two membrane here. It's between the membrane here, right? So this gap junction will act as a communication medium. Okay, communication medium. And what all it transfers, it can be ions or it can be molecules or it can be impulses. Okay, now you understand what is the function of the gap junction. Now let's move to the next question here. Ball-Biani rings are sites of dash. Okay, this question was again asked in the year 2015. Okay, fine. What do you mean by these Ball-Biani rings? Have you ever heard about it? See, they are large puffs. Okay, they are large puffs of polytene chromosomes in the ditryptane. Okay, it belongs to some organism called as tentans. Okay, it is present in tentans, and these puffs are particularly well suited for studies of the structure of the active genes and the synthesis and transport of specific RNA. Okay, a specific RNA protein. So RNA and protein synthesis will occur in Ball-Biani rings. Okay, so Ball-Biani rings are the sites of RNA and protein synthesis. It is option C, which is the right answer. So these are nothing but puffs. If you see the if you see the diagram, you will understand. You have a picture which shows a clear image of the Ball-Biani rings. It's actually a chromosomal puff which happens there. Okay, so this region, can you see here? It's a large puff, right? It's a large puff that happens, right? They are well suited for the studies for the structure, when you have to study about the structure of active genes, the synthesis that takes place, the transport of proteins, all that will come under that. Okay, so therefore the correct answer to this question is option C. Now let's move to the next question here. This question was again asked in the year 2015. Okay, you need to match the columns and identify the correct option. Okay, you have different different terms here like thylakoids, cristae, cystine, chromatin. Now let's go one by one, right? So what do you mean by thylakoids? We've already seen in the earlier parts, right? We have already seen in the earlier parts. If you remember, thylakoids are flat membranous sacs in the stroma. Therefore the first one, thylakoids, belong to the answer third one, this one. Okay, here third one, flat membranous sac in stroma. Now if you take cristae, cristae is, any guess? Cristae are the infoldings that are present in the mitochondria. Okay, so the correct answer for this, see, thylakoid comes here. I'm sorry, thylakoids will come here. Okay, and this part, if you see, cristae belongs here. Okay, now coming to what is cystinae. What is cystinae? I think it should be easy now. Cystinae are the disc-shaped sacs that are present in the Golgi apparatus. Okay, and last one, chromatin, you know, it's the condensed structure of the DNA. Now you understand what these four terms stand for. Thylakoids are the flat membranous sacs in stroma. Cristae are the infoldings that are present in the mitochondria. Cystinae are the ones that are disc-shaped sacs present in the Golgi apparatus. And chromatin is the condensed structure of DNA. Therefore, the correct answer is option A. Okay. Now you understand? Fine. Now let's move to the next question here. This question was again asked in the year 2015. Cellular organelles with membranes are dash. Okay. Right. Now coming to the concept. Cellular organelles with membranes. We already saw in the earlier parts there are organelles with single membrane, with double membrane, or no membrane, right? We have seen single membrane, double membrane, no membrane. So any example for the single membrane? See, single membrane is present for organelles like lysosomes, vacuoles, and double membrane is present for mitochondria, chloroplast, Golgi apparatus, nucleus. And when I say no membrane, you can call it as a membrane also, an organelle without a membrane. Okay, ribosomes, centrioles, and nucleolus will come under no membrane system. Now I have almost given the clue. Therefore, the correct answer goes to option C, lysosome, Golgi apparatus, and mitochondria. Because in the first one, if you see, they have spoke about ribosomes. I told you ribosomes are membraneless organelle. Therefore, that will also not come. Option B, you have nuclei, which will not get that. And option D, you have the same problem with nuclei and ribosome. Therefore, the correct answer is option C. Now, let's move to the next question here. Read it here. This question was asked in the year 2014. The solid linear the solid linear cytoskeletal elements having a diameter of 6 nanometer and made up of a single type of monomer are known as dash. Okay. So here, if you see, we are talking about the cytoskeletal element. Okay, they've given you a clue. And this question we've already again discussed in the earlier parts, where we spoke about microfilaments or microtubules is the answer. The correct answer goes to microfilaments, which is responsible for the structure, for the overall background of your cytoskeleton. So here the question is very clear, where they talk about the cytoskeletal elements which have a diameter of 6 nanometer and they made up of single type of monomer. Monomer means single chain. Single. Okay. So microfilaments are cytoskeletal elements which are about 3 to 6 nanometer in diameter, and they are made up of a protein called as actin. Now, what is a microtubules made up of? That too has a protein in it, and that protein is called as tubulin. Okay, therefore the correct answer to this question is option B, microfilaments. Okay, now you can see a diagram to your right side which talks about microfilaments and microtubules. Now microfilaments has a protein which is called as actin, and microtubules will have tubulin. Okay, that will be the protein associated with it. Therefore, the correct answer is option B. Now, let's move to the next question here. This question was asked in the year 2014. The osmotic expansion of a cell kept in water is chiefly regulated by dash. Okay, so coming to this question here, what do you mean by osmosis? How can you say, or how can you describe about osmosis? Is it lower to higher concentration or higher to lower concentration? See, here they are talking about the osmotic expansion of a cell. Okay, and that too when it is kept in water, what happens there. Now for example, let's take sponge here. Okay, let's take sponge. So what happens when you take a sponge and you keep it in the surface of water? What does it do? What does a sponge have initially? It will have small holes in it where it will start to absorb water. Likewise, the osmotic expansion of the cell kept under a water is chiefly regulated by the vacuoles that are present in it. As a sponge has small holes in it, now these vacuoles are responsible for this expansion when it comes to a cell. Okay, the example I think made you clear what it is. So now if you see, this vacuole is bound by a single membrane which is called as tonoplast. Okay, so it is here. It is called as tonoplast. It also functions as a semi-permeable membrane. What do you mean by semi-permeable? Selective. Okay, selective. This means selective. That is, they will not allow the entry of every molecule into their system or any ions into their system. They will selectively send only a few which is nearly necessary for the system. Okay, semi-permeable is the meaning of that. It segregates the vacuolar contents from the cytoplasm and allows the osmotic entry or exit of water. Okay, therefore the correct answer to this question is option B, vacuoles. So you have a diagram which shows you a vacuole and you have the tonoplast which is around it, right? So tonoplast is a single membrane that covers a vacuole. Now next question. This question was asked in the year 2014. Match the following and select the correct answer. Once again, it's a match. Sometimes there is a possibility for you to get questions like this, match the following, or sometimes they'll give you diagrams and they will tell you to mark the parts. So these questions, they actually not only test your knowledge, but they also test how much you have undergone or how much in depth that you've gone with this subject. So it will tell you that. So now if you see this match the following, you have 4 terms to your left and 4 terms to your right. Right. Okay. Now coming here, the correct answer to this question. Now what do you mean by centriole, chlorophyll, cristae, ribosomes? I think it should be easy for you. We'll go with the ones that we know first. Chlorophyll. Chlorophyll is present in dash. Cristae, we already saw it is the infoldings in mitochondria. Ribosomes, they are associated with. Any idea? See, I'll give you the answer for this. The correct answer to this question is option A. Okay, centriole is the basal body of cilia of flagella. Okay, and centriole, if you see, it forms actually centrosome is an organelle which usually contains 2 cylindrical structures called as centrioles. So they help or they form the basal body of the cilia and the flagella. Okay, cristae, as we already saw, chlorophyll, they are present in thylakoids. Cristae, we already saw infoldings in the mitochondria, and ribosomes, they are present in nucleic acids. So this question is very easy. See, whenever you get a match the following, always look into your left side first. If you have any terms which is familiar to you, you can start finding in that order. Now, as I already gave you a hint how to do it. See, when I gave you these 4 options, cristae, we've already discussed what it is. So cristae, infoldings in mitochondria. So already we have cancelled out 1 option. So the remaining 3 options you have to fit in the other 3. So this is how you should do a match the following when you are not sure about the answers. Okay, now let's move to the next question here. Yeah, I just told you there are chances for you to get questions when the situation is where they will ask you a diagram and they will ask you to mark parts, or they will show you the diagram and they'll ask you what organelle is it. So this question is similar to that. It was asked in the year 2013. Which one of the following organelle in the figure correctly matches it with the function? Okay, first you need to identify what organelle is this, right? It is very important that you identify it first. So any idea what this diagram is or this picture is? It's the rough endoplasmic reticulum. Okay, as we all know, endoplasmic reticulum can be smooth or, it can be smooth or rough. So it can be rough endoplasmic reticulum or smooth endoplasmic reticulum. Now this scene, this particular picture represents the rough endoplasmic reticulum, and you all know the smooth endoplasmic reticulum is responsible for an activity. Likewise, the rough endoplasmic reticulum is also responsible for another activity. Likewise, the rough endoplasmic reticulum is responsible for protein synthesis. Right? They are responsible for protein synthesis. Therefore, the correct answer to this question is option C. Rough endoplasmic reticulum is responsible for protein synthesis. They are extensive and continuous with an outer membrane of a nucleus, and this endoplasmic reticulum, or it can be called as a network, which is actually scattered part in the cytoplasm. Okay, now let's move to the next question here. This question was asked in the year, again, 2013. The Golgi complex plays a major role in dash. Okay, now coming to this question here, what is the major role of the Golgi complex? Okay, now what is Golgi complex? Before we go into the answer directly, we should know what is Golgi complex or Golgi apparatus, whatever you can call it as. Now, this plays a very major role in many activities. Okay, Golgi body or Golgi apparatus, it plays a major role in many activities, sometimes in post-translational activities, and this Golgi complex can be described as a complex of vesicles, okay, and folded membranes within the cytoplasm. So where they are present? Within the cytoplasm, and most of the eukaryotic cells or eukaryotic organisms which are involved in the secretion and intracellular transport, Golgi complex will play a major role in it. Okay, therefore the correct answer to this question is it helps in the post-translational modification of proteins and glycosylation of lipids. The correct answer is option C. Okay, see, to your right side they have explained you the reason. What happens here is it plays say post-translational. What do you mean by post-translational? It is after translation happening. As we all know, a eukaryotic organism will undergo transcription and translation. Likewise, this plays a major role in post-translational modification of proteins, forming glycoprotein and glycosylation of lipids, which form glycolipids as a result. Okay, so therefore the correct answer is option C. Now let's move to the next question here. This question again in the year 2013. Easy question. The major site for synthesis of lipids is dash. We already saw the endoplasmic reticulum is divided into rough and smooth, and we all know the rough part is responsible for protein synthesis. So how about the smooth part now? It is responsible for lipid synthesis. Therefore the correct answer to this question is option A, smooth endoplasmic reticulum. Okay, in animal cells, lipid like steroid hormones are also synthesized in the smooth endoplasmic reticulum. This question is very easy. I think by now you have guessed the answer. To your right side you can see the picture which represents the smooth and the rough part of the endoplasmic reticulum, and the activities are really unique. Both have different activities. Now let's move to the next question here. This question was asked in the year 2013. Again 2013. Okay, let's read it here. Which one of the following type of plastids does not contain stored food material? They're asking which does not contain. Okay, now coming to each option here. You have amyloplast, lyoplast, chromoplast, uroplast. So what is the correct answer here? Any idea? The correct answer here is option B, chromoplast. Okay. Now this chromoplast, if you see, they are non-photosynthetic colored plastids which synthesize in the store of carotenoid pigments. See, chromoplast is responsible for the color. Okay, they are not responsible for the storage of food material. Okay, now you understand. See, each one has an activity here. For example, if you take amyloplast is responsible for the storage of starch, as you see here. Then neuroplast stores proteins. Leoplast stores oil droplets and fats. Therefore, chromoplast, which is responsible for the color or the pigment, does not store any food. Therefore, the correct answer is option B. Now, let's move to the next question here. Again, this question, if you see, they have asked you to mark the part A. So, as I already said, you might get questions like this, which will test the depth of your study in your book. Okay, therefore, now let's read it here. Select the alternative giving the correct identification and function of the organelle A in the diagram. Okay, fine. Now this diagram shows you a picture, and the options are endoplasmic reticulum, mitochondria, Golgi body, lysosomes. So any idea what it is? The correct answer goes to option B. So they have clearly marked this region which shows mitochondria. Right, this picture clearly shows mitochondria. So mitochondria produces cellular energy in the form of ATP. Okay, therefore the correct answer to this question is option B. Okay, so these questions can be expected in NEET. It is very important that you go in depth with your book and study. Now let's move to the next question here. See, this question was asked in the year 2012. Okay, let's read it here. Ribosomal RNA is actively synthesized in. So where is RNA, ribosomal RNA synthesized? I think by now you're all guessed what the answer is. Yes, the answer is ribosomes. We all know ribosomes is a factory for RNA, right? Ribosomal RNA, which is also called as rRNA. We always resemble in that form, right? They are molecules in the cells that forms a part of the protein synthesizing organelle, which is known as ribosome, and they are exported to the cytoplasm. And what do? They help in the translation process. That is, they translate the information in mRNA into protein, and this rRNA is actively synthesized in the nucleolus. Nucleolus is also called as ribosomal factory. Therefore, the correct answer to this question is option D, ribosomes. So here you can see the nucleolus, as I already said, nucleolus, the other name is ribosomal factory. Okay, ribosomal factory. Therefore, ribosome is a place where ribosomal RNA is actively synthesized. The activity is that it will translate the information into mRNA. mRNA is nothing but the messenger RNA, right? Therefore, the correct answer is option B, nucleolus. Okay, so nucleolus or ribosomes, they all mean the same. Okay, you don't have to get confused here. Ribosomal RNA, molecules in the cells that forms a part of the protein synthesizing organelle. So here, if you see, they are talking about the part or the organelle which actively synthesizes ribosomal RNA. So rRNA, if you see, it is present in ribosomes, but where it is synthesized, if you ask me, it will be synthesized in the nucleolus. The other name for nucleolus is also ribosomal factory. Therefore, the correct answer to this question is option B. Next question. Again, this question was asked in the year 2012. What is true about ribosomes? I think you'd have guessed the answer by now. Yes, the correct answer is... So what is ribosomes first? Do ribosomes have membrane in them? No, ribosomes are membraneless. They do not have any membrane. They are an organelle which are composed of rRNA and proteins. So where are they present now? Are they present in prokaryotes or in eukaryotes? This ribosome, if you see, they are actively present in both. What is it? Prokaryotes and eukaryotes. Okay, prokaryotes and eukaryotes. Right? And another thing is that in prokaryotes it will be usually 70s subunit, and in eukaryotes it will be 80s subunit. Okay, this is the extra information that you need to know. Therefore, the correct answer to this question is option B. These are composed of ribonucleic acid and proteins. Okay, next question here. So this is the last question for this part 3. So let's read it here. Select the correct statement from the following regarding the cell membrane. So this question was asked in the year 2012. Okay, let's go one option by option. Look at the first option here. You have sodium and potassium ions move across the cell membrane by passive transport. We all know when it comes to transfer, I mean when you come to transport system, you have two types of transport system. One is active and the other is passive, right? The next option, if you see, proteins make up to 60 to 70% of the cell membrane. And option C is lipids are arranged in a bilayer with polar heads towards the inner part. So here they are asking us the correct statement regarding a cell membrane. Okay, the correct answer is option D. Fluid mosaic model of cell membrane was proposed by Singer and Nicholson. Okay, so now tell me, what is this fluid mosaic model? It describes the structure of the, what does it describe? It describes the structure of the plasma membrane, right? It describes the structure of the plasma membrane. And here if you see, the components are actually expressed in the mosaic model. Okay, so here it describes the structure of the plasma membrane as a mosaic of components, which includes, what are the components that are present there? Phospholipids, right? Phospholipids. What else? Cholesterol, right? Cholesterol. You have protein, you have carbohydrates, right? So these are some of the components that are present. So these components will represent in the mosaic form. Plasma membrane, if you see, it will range from 5 to 10 nanometer in thickness, and this model of fluid mosaic membrane was actually proposed by Singer and Nicholson. So out of the 4 options, the correct option is option D. So by now we have completed part 3. Thank you for watching the video. We will all meet in the next part of the same chapter. Thank you again.